So dear students, we have with us Dr. Bhattacharya. He is working as a scientist in mycology and microbiology department, totally in Research Institute, Tea Research Association, Jorhat, Assam. And he is working there since 2014, so it's been more than 6 years now. And earlier he was a research fellow at ICAR during 2012. He is a two-time gold medalist during his bachelor and master from Guwahati University in 2004 and 2006. He is a recipient of many awards and honor, like National Merit Scholarship, Gold Medalist from University, Professor A.C. Datta Memorial Awards, Best Researcher Award from the International Scientist Award 2020 on Engineering, Science and Medicine by prestigious Pretty Good Professional Association India Sadhana Young Achievers Award, BioMed Young Scientist Award. He is also annual member of Association of Microbiologists of India. He has more than 40 research publications in national and international rapid journal. He has also published 2 books on microbes, and he has guided many graduate and undergraduate students. He is also heading many institutional projects as well as funded projects from Government of India as a PI and co-PI. And he has a vast experience working in microbiological research. So with this very brief introduction, on behalf of Srishti, I welcome you, sir. Over to you.
Now your slides are visible, na?
Sir, I'll just do it.
Okay. Okay. So good morning, my dear students, and the organizer for introducing me and for providing such an opportunity to give a lecture before the students. It's always a privileged matter to give a lecture before the students. So please, ma'am, as it is not happening from my side.
Yes, I'm doing, sir, but I think I have some issues. Can you see the slides?
No. Dear students?
No, something is wrong.
Oh,
sir, can you stop sharing because we
okay.
Can see.
Okay. Stop sharing. Yes.
Thank you, sir.
Yes.
Yes, sir. I'll do that now.
Yes, I think now it is visible, na?
Okay, sir.
Yes, now it is visible. My dear students, is it visible?
Yes, sir, it's visible.
Yes. So, ma'am,
Yes,
may I start my lecture?
Yeah, you can start, sir, and please instruct me whenever you want to go ahead.
We have to complete this session within 1 hour, no? So now it is 55 minutes to go, I think.
Yes.
So please come to my presentations. So I am Pranabhananda Bhattacharya, so doing as a microbiologist in the Tuklati Research Institute. So today's topic is concentrated on the use of microorganisms and their role actually for decomposition of agricultural wastages. So as I am working in the Tea Research Institute, so I have also included a part of tea to give a example where we can use these agricultural wastages as a role model in tea ecosystem. Okay, so now let me illustrate my points or the slide shares for your kind concern. So as you know that agriculture is one of the important, means it is the bone of any kind of organisms. Na? So these agriculture required nutrients. This agricultural system, it may be either a food, means fruit yielding crop, or it may be either a any kind of vegetables or any kind of horticultural crops, or it may be a non-beverage yielding crop like tea. So everywhere these requires actually nutrients, and the nutrients, they may be either of different categories. So some nutrients are required, you know, that in major amounts like carbon, NPK, nitrogen, phosphorus, potassium, and there are also other nutrients which are also required in some small amounts or needed in a very trace amounts. So overall, these all nutrients, they make up the bone of this agriculture. Without the support of these nutrients, agriculture cannot run. So now, please, ma'am. Next slide. Yes. So this is the table showing the importance of these nutrients for different kinds of plants. As you know that nitrogen, so it is required for protein development or building blocks of the proteins, then nucleic acids, etc. Then phosphorus is required for nucleic acid formation. ATP means energy formation, then phospholipid formation. Then potassium is required for enzyme activities. And then lots of other nutrients have also other kinds of mechanisms, as a result of which entire agricultural system grows well. However, there are lots of mechanisms for uptake of these nutrients. You know that roots are the basic, means organelles, or means they are structures by means of which these nutrients are absorbed from the root, means by the from the soil in incorporation with water. Means water plays an important role while providing the nutrients to the plants, growing plants. So there are lots of mechanisms like physiological processes, mass flow happens, diffusion happens, interception happens. I will not go in detail about these mechanisms, but these are different mechanisms by means of which these nutrients are absorbed by the plants. Next slide. Next slide, please. Yes, so you know photosynthesis is one of the vital processes for plant growth and development. For this also nutrient is required. So carbon, and then in presence of this atmospheric carbon dioxide, then water molecule, you know, this type of photosynthesis means sunlight is essential, then chlorophyll is present in plant leaves, and ultimately this glucose production takes place. And this glucose serves as the main food for almost all kinds of plants and animals. Next, please. So as nutrients are required for growing plants, there are also different nutrient deficiency syndromes. So as these nutrients, if they are not available to the growing plants, suppose nitrogen, phosphorus, potassium, so these are some of the macronutrients. Then there are also some micronutrients like zinc, boron, manganese, magnesium. So with the deficiency of their required load, the plant suffers from different kinds of deficiency syndromes. As you have heard, or as you know, that there are lots of deficiency syndromes like your necrosis, chlorosis, leaf veining. Okay, so there are lots of damping of these kinds of diseases. So these also develops due to the plants when they are getting in stress actually. So nutrient deficiency in plants is also one of the important fact for which nutrients are very much more essential in their required doses. Next, please. So that's the point actually. So nutrients are required for the plants. Okay, so these nutrients are basically present in native soil. Soils naturally contain many nutrients like nitrogen, phosphorus, calcium, potassium. Okay, but what happens due to the long-term cultivation of the crops, or the crops growing for years after years, these nutrients gets depleted in the soil, as a result of which there is a refilling of soil nutrients. Means there is every need for refilling of soil nutrients, and this refilling of soil nutrients is usually done by the act of fertilizers. Okay, so this is the point why we need to apply the fertilizers in soil. So fertilizers are simply nothing but these are simply plant nutrients applied to agricultural fields to supplement required amounts or elements, required amount of elements found naturally in the soil. So they have to give the nutrient, means deficiency, solve the nutrient deficiency related problems in soil. Next slide, please. So These fertilizers, they may be of different types again. So there are chemical fertilizers and there are some natural fertilizers. So chemical fertilizers, they may be either of nitrogenous fertilizers, phosphatic fertilizers, they may be either potassic fertilizers. So these fertilizers are usually added to the soil in the form of different compounds like urea, ammonium nitrate for nitrogenous fertilizers, then rock phosphate, superphosphate for phosphatic fertilizers. So then muriate of potash, sulfate of potash, these type of fertilizers are usually added, or they have been long been practiced in agriculture. Even you will surprised to know that application of chemicals are in practice in agricultural systems since 4,500 years ago. So it's a very long back, no? So we are applying chemical fertilizers for the improvement of plants. Besides these chemical fertilizers, there are natural fertilizers like soil amendments. So gypsum, then limestone. So these are generally added to improve the soil textures related properties like your porosity problems, then air means aerosol problems. So these type of problems can be means soil amendment means these are some of the important materials for nourishment of the soil, or you can simply call as the conditioner of the soil. So besides this, our today's presentation is basically concentrated for the use of organic fertilizers. Why we should go for decomposition of agricultural wastages, or why should we apply those agricultural wastages for the decomposition by some biological agents. I will go in detail in next slides. So for the production of some manures or composts, okay, so what is the need for them, why they are advantageous than to these chemical fertilizers? Next to presentation. So this is another thing. I'll just mention that these pesticides have also been long been used in different agricultural system, not only in tea. Everywhere the pesticides have to be applied, so they can be considered as actually as inessential evil. So these pesticides, they can also be falls under two categories, basically chemical pesticides and then to your biological pesticides, means biopesticides. So chemical pesticides, they are, you know, everywhere you can now see that chemical pesticides are they have lots of harmful effects. So what are those harmful effects? I will show you. But biological, that's why we have to move our eyes, or we have to move our concentrations. The global demands for application of some biological substances for non-chemical sustainable productivity. Next slide, please. So this is the chart showing lots of disadvantages using the chemical means applications or chemical substances in agriculture. As you know, after the application of chemicals, so it plays an important role in causing pollution of air and groundwater, as volatilization, leaching activities, this regularly happens in soil, as a result of which the applied chemical goes back to the atmosphere, or either to the soil or either to the water bodies, as a result of which water bodies also gets nutrient rich conditions. In several cases, you know that it is called as the eutrophications. Due to application of large amount of pesticides or injudicial and then injusticiable amount of means pesticide application or chemical applications in soil, they causes soil contamination. Soil contamination means there are deposition of large numbers of emerging contaminants, heavy metals, this type of thing. So as a result of which soil quality deterioration takes place, and this is linked to fertility loss, land degradations. Then it also reduces the biodiversity. You know biodiversity. So diverse organisms like natural flora, fauna get depleted, including our most important beneficial resources, means microbial resources. These microbial resources, suppose the microbial resources, as for example, your soil microbial resources. You know that soil microbial resources are very much more beneficial for us. Okay, so they plays an important role while making available the inorganic nutrients of soil to the available, means inorganic nutrients which are unavailable to each available form, so that plants can absorb them and get there from nutrients. Okay, so due to application of chemicals, what is happening? These beneficial microorganisms gets depleted from our agricultural field, then reduces nitrogen fixation abilities of the plants and then other nutrients. Okay, then pollinator decline. You know the importance of pollinators. They play a very important role in different fertilization activities. So these pollinators get dead and they are getting declining, or their populations is getting declined day by day. Besides them, there is another set of organisms that is known as natural enemies. These natural enemies and then butterflies, they get depleted from our ecosystems, natural ecosystems, or our agricultural fields, or our tea systems. So as a result of which there is the occurrence of lots of pest problems. So natural enemies and butterflies, they eat lot number of pests in agricultural system. So due to their depletion in numbers, so these pest populations becomes very increased in nature, then threatens our endangered species. Then it also another important problem is that chemical resistance development, means development of chemical resistance in diverse pests and pathogens. This means that earlier what those chemicals which have been applied for controlling some pests and pathogens, they worked well, but nowadays they are not working well because they are developing resistance to those chemicals because they have been applied long back. So this is another very alarming effect to agricultural system. Then disease development in plants. There are lots of diseases in plants which generally happens due to the degradation of land systems or due to soil deterioration or fertility loss conditions, because as your fertility is lost, the plants get stressed, and the stressed plants naturally invite those kinds of pathogens. Next slide, please. And overall, this chemical pesticide or chemical supplements application in any kind of agriculture is also not cost effective. So this slide shows us application of chemicals, lots of chemicals which have harmful effects. They are causing deterioration in our natural ecosystem environment, causing the pesticide cycle. You know pesticide cycles. These pesticides we apply in soil, but they are getting volatilized or leaching activities to nearby water bodies, and ultimately they also goes due to volatilization or vaporization to our atmosphere. Precipitation comes, and these again get back to our soil conditions. So what is happening? So we are getting back by these harmful effects of chemicals on a regular basis. Next slide, please. So persistence, bioaccumulation, and biomagnifications. These are some of the harmful effects of these chemical supplements which are we regularly used in agricultural systems. So as a result of these harmful activities, our food chain getting disrupted, or there is an imbalance in ecosystem, entire ecosystem, our food webs getting disturbed. Lots of animals, you have also read in different newspapers or your books that lots of animals which are come to our places, or lots of animals these are earlier in our places, but now they are getting to their extinction or they are getting endangered. Okay, so this is one of the reason for the application of this persistence, bioaccumulation, and biomagnification activities of these harmful chemical substances. Next slide, please. Next slide, please. Yes. So ultimately, this is another problem, change in climate, global warming, greenhouse effects. So these chemicals, when they get back to the atmosphere, they are also another problem. We all are now threatening of these problems. So due to this global climate change, or due to these abrupt changes in our climate, so everywhere, not only in agriculture, our human populations, animal populations are also getting affected with slowly in a rapid manner. Next slide, please. So now, in context to tea ecosystem, what are the damages caused by the chemicals? So this is just a role model to show the activities of chemicals, what they are doing actually in our agricultural systems. So soil degradation or loss. So this is one of the important activities. Soil degradation means their quality is getting deteriorated due to application of injudicious and injustifiable application of these pesticides or these different types of chemicals. Soil salinity is affected. Then localized waterlogging problems. Localized waterlogging problems is also another important problem in the agricultural systems, as a result of which the plants gradually losses their abilities to absorb their nutrients, because they are unable to absorb their nutrients, because these chemicals are causing their problems in their root hair size, their root length size, their porosity sizes, etc. Then fertility loss, deteriorated soil normally lose their fertilities. Then predators, I have mentioned that predator loss, and then natural enemies loss, contamination of the water bodies, lots of substances, unwantable emerging contaminants are now everywhere in our ecosystem, in our agricultural sectors. Then plants getting stressed, means whenever the plants cannot absorb their nutrients well, whenever there is waterlogging problems, whenever there is a fertility loss, automatically the plants get stressed, and stressed plants normally invites different kinds of pests and pathogens. Another thing is that resistance development of pests, creation of undesirable residues, MRL problems Have you heard? I think you have heard the MRL related issues. Means we have everywhere for application of chemicals there is a judicial body, so we have to use a means in a standard limit of actually chemicals, so that is our residual limit. Whenever it crosses to the residual limit, it is known as maximum residual limit, and these kinds of problems now arises in everywhere. So met contamination in context to tea ecosystem and residue problems. There are also problems related to soil ecosystem or tea ecosystem or agricultural sector, like soil compaction, soil getting very rough, they are very getting hard, as a result of which plant roots cannot absorb more amount of nutrients, or whatever the required amount of nutrients, they are unable to absorb them. Erosion activities, land degradation. So these all happening due to the application of the chemicals. Lot of other problems are also related, but chemical is one of the important factor while causing this land degradation. Then soil means infertility related problems. Next slide, please. So this is, I am showing one of the T-bows. No? So this is one of the model system. So where what is happening, this T-bow is getting affected. I am representing here that this plant is in stress. Whenever it is in stress, there is a adoption or there is a invitation of lots numbers of pests and pest problems. So plants getting in stress. Next slide, please. Next slide. Yes. So injudicious and illegitimate application of chemicals are harmful. Why harmful? Because they are causing the plants in stress. And the stress problems we have noticed in our tea system. As a tea microbiologist, I have noticed that there are lots of diseases. Some portions are not coming in the lower side. So red rust and violet root rot. So these are some important secondary diseases in plants which regularly comes due to stress related problems in plants. And one of the important reasons for stress is chemical application. That means we are destroying our environment with the injudicious and illegitimate application of chemicals. Besides, waterlogging is another problem which is frequently observed in tea growing areas. So this waterlogging is also another important causes of land degradation. Next slide, please. So we have to move towards sustainability. Without having sustainability or without having some non-chemical approaches of this nutrient management, we can't move forward. We can't move forward with our young generations. So we have to go for some organic approaches. We have to go from our sustainability approaches. And what to do? We have to legitimate application of chemicals. One day we can't replace the chemicals. We have to reduce their limits. By how? We have to develop some integrated nutrient management approaches, or we have to develop, in case of disease suppression, we have to develop some integrated disease management approaches. So here we have lots of substances in our agriculture, which is known as commonly agricultural wastages. Next slide, please. Next slide, please. Yes, so United Nations in 2015 have also identified 17 different goals for sustainability development. So we have to reduce the load of chemicals. Otherwise, we can't survive in this peaceful earth. No? So we have to reduce our chemical load and we have to go for sustainability approach. Next slide, please. Next, please. Yes, agricultural sustainability. So what we'll do now? So we need nutrients. As you have already learned that agriculture needs nutrients and these nutrients have to be supplied. Okay, because there is a gap in the nutrients in the every ecosystem or every soil conditions. But where from nutrients? Where we can get these nutrients? So we have to do some non-chemical sustainable approaches for achieving those nutrients. Kinetics in organic matter decomposition using an eco-friendly and sustainable approach is needed. Why? To grow the farmers, to mindset, to means improve the mindset of the farmers worldwide, to leave opportunities for the use of non-chemicals against toxic chemical supplements. We no more apply chemical toxics. No? So we have to do some alternative approaches. We have some cellulose degrading microorganisms. I will now explain how these cellulose degrading organisms or how these microorganisms, they have to put their work for the decomposition of the agricultural wastages. Besides these cellulose degrading microorganisms, or you can simply call this cellulose degraders or cellulolytic microorganisms, there are also some potent worm species, means there are some earthworm species which can be compiled together, which can be co-inoculated together for better productivity or for better production of our organic manure, which can be applied in the agricultural system for better compost recovery, nutrient uptake, and then maximize the load of microbial, that means those beneficial microbial populations like nitrogen fixers, phosphate solubilizers, potassium mobilizers, because until and unless the microbes are at work, we can't get a good agricultural productivity or we can get a good soil health conditions. Next slide, please. Next slide, please. Yes. So you know agricultural wastages, there are lots of fruits, there may be lots of vegetables in your kitchen garden. There may be lots of agricultural wastages, okay? Abundance of natural plant residues, fruits and vegetable wastages and weed biomasses, okay? In and around the farmlands, they can serve as the agricultural wastages. They are the excellent source of nutrients. But if used properly, if we use these agricultural wastages, they can be converted to some nutrient-rich components and they can be then applied to our agricultural system. And this is the now strategy for sustainability development in agricultural system. Without these sustainability practices, our agriculture cannot run more. So in case of the ecosystem, if you simplify this as a role model, so we have lots of actually agricultural means they have lots of wastages actually. So we can use them, rice straw bar, waste tree loaves, weed biomass, pruning litter, decomposed wood nearby areas, water hyacinth. So all these substances can be collected and they can be processed for composting using these what cellulolytic microorganisms or some this type of degrading organisms, biological agents along with earthworm species. Next slide, please. Yes. So using this microbial approach actually, so we have to know what is cellulose. You know cellulose. Cellulose is a linear polysaccharide. Okay. So most of the agricultural residues, plant biomass, weed biomass, they contain about 40 to 50% cellulose. So this cellulose, if they can be converted, they can be finished to some end products, very simple products, which can be easily utilized by the plants. Besides, due to these composting activities, or simply it is called as biodegradation activities, okay? Biodegradation or biodecomposition activities by these beneficial microorganisms to these agricultural wastages, okay? So their nutrient get enhanced because there is a decomposition activities. These microorganisms have some enzymatic activities by means of which they can convert this inorganic locked inorganic nutrients to the organic form. So you can see some of the examples of cellulolytic hydrolysis. These are not actually, these have been collected. So these are some hydrolysis activities just to show you. So these hydrolysis zones, I marked these as a cellulose degraders. So these are the basic characteristics of growing in culture place. So next slide, please. So what are those cellulose degraders? So cellulose degraders, it may be a bacteria, it may be a fungus, it may be actinomyces, and more recently, in the last 2 decades, so we are also working, entire world is working with some cellulose degraders, which is known as microbial consortium. So what is microbial consortium? This microbial consortium is nothing but this is the compiled effort of this bacteria, fungus, and actinomyces, those which can grow in a very compatible manner. So those microbes which can grow in a compatible manner in your in vitro conditions, they can be used as microbial consortium for their decomposition activities of agricultural wastages. Okay, so this simply cellulose degraders, what are these? Microorganisms that can produce cellulose enzymes. Lots of microorganisms have their establish their efficiency in cellulose degradation, like your Bacillus, Pseudomonas, Serratia, Trichoderma, Feriformospora indica, Penicillium, Aspergillus, Homa, Fusarium oxysporum. We at Tuklai, our department, we are also doing some of the important strains I will show you. So of this microbial decomposition activity, so that these agricultural wastages can be utilized in a proper way for the quality organic manner, so that we can get better nutrient recovery percentage, nutrient uptake, and then increase the beneficial microbial load in agricultural soil, particularly this tea soil ecosystem. Next slide, please. So this is the one of the important mechanism by means of which cellulose getting degraded to simpler products and the plants can absorb them. This is in the left side, you can see the cellulose degrading microbes growing. in their plates like cellulose agar media. I will show the media. And then this is the confirmatory test by means of which the cellulose degrading microbes, you can confirm them for their isolation. Next slide, please. Yes. So how to collect them, how to isolate them in your media or in your laboratory? So you have to first isolate them. You have to first potent screening before going for agricultural waste decomposition. We have to have the decomposers. No? So for this isolation of this decomposition, isolation of these decomposing microorganisms, it is very first prime requirement. So there are lots of media. So we are doing with these medias our cultures. So Mendel-Rees media, then CMC agar plates, cellulose agar media, so for their better growth. So in the right side, in the topmost corner, you can get the hydrolysis activities of our strains. No? So besides, after growing them, we have to do their pathogenicity test. And this pathogenicity test is very much more important, because until and unless the microbes are non-pathogenic, we can't have their means application in agricultural system, because for this reason, we have to have the pathogenicity test for the isolated microorganisms. This is a prime most importance for use of their decomposition activities in agricultural wastages. Next slide, please. So after this, so molecular characterization is also another demand for quality identification and then proper identification, and to trace their evolutionary relationship. It is very much more essential fact. Then nowadays, as metagenomics is also doing very good, because you can unlock the massive microbial diversities, okay, so present in the environment, soil, water, for novel isolation of novel molecules. So next slide, please. So this is one of my own publications which I published on this metagenomics. So this metagenomics is very important tool for direct extraction of the microbial gene pool or the species level informations, and work with this as a whole in our decomposition activities. So next, please. So filter paper degradation assays, this is another tool, or this is another technique by means of which we can get confine, or we can get confirm the cellulose degradation activities or abilities by our isolated strains. So here we have to have always do some control experiments. No? So the degraded parts you see in the figure, so these are the degradation activities done by the cellulose degraders in the laboratory conditions. Unless and until you confirm their activities of this cellulose degradation in your lab, you have not to pass to their agricultural wastages. No? So this is first time of confirmation of their growing in the confirmatory medias, and then see their degradation activities. If the microbes are going positively for their degradation, so you can use them for further experimentation. Next slide, please. So significant achievement was obtained when we got the, or when we done the experiments, we have achieved a significant achievement in NPK against control. Means agricultural wastages, when we have processed by microbial degradation activities, and when we have applied in soil, we are getting more amount of nutrients than those conventional systems. No? So microbial populations, including those like phosphate solubilizing microorganisms, nitrogen fixers, beneficial or antagonistic actinomyces, even M fungi, and then cellulose degraders also getting increased due to the application of this biodegraded material. Next. So we have applied in T system. So we are growing the chalas with this compost in sleeves, and we are getting a very good, healthy root formation against control. Next slide, please. This is another, we are practicing in different clones. I think you have heard about different clones, TV clones or T clones actually. So we have practiced this compost application in different clones to get their results against control. So we have got a very significant compost recovery, then nutrient uptake, and lots of other beneficial microbial inputs. That means they have been increased with the application of this biodegraded materials, which is done with the help of these decomposing microorganisms. Next slide, please. So we have done some field experimentations. Right side, this is the mass cultivation. When you have to go for field applications, you have to do the mass cultures of these microbes. And these microbes in the right left, means in the right bottom side. So these are the cultures of microorganisms. They can be used to decompose the agricultural wastages. And as these microbes have some I have already mentioned the enzyme means production capacities, so they can degrade these agricultural wastages to quality compost manner, and we have applied in the agricultural field. So after application, we are getting a very good response of the plants regarding their soil health, plant growth and productivities. Next slide, please. So bioconversion of organic residues for their recycling, this is one of the important assets. Recycling means we have to nutrient recycle, and with the help of these cellulose degrading microbes, we can recycle for improving the strength of organic nutrients. I have already mentioned that toxic chemicals in agriculture are already deteriorating our environment physically, chemically, and biologically. Next slide, please. So these are some of the experiments or observations where we can also use different other wastages, agricultural wastages, including the tea wastages, those tea materials or tea products which are not can be used by our human beings. They can also be decomposed by these cellulose degrading microbes. You can see the color difference. No? In control, you can see the control color and you see the compost color. So that means after degradation their color also getting changed. This is one of the important indicator of quality compost. Next slide, please. So CDMs, more about them. So they produce different enzymes. They play an important role in potential decomposition of agricultural wastages, as a result of which their work has been established for enhanced organic matter decomposition. Okay, so they have rapid growth. Why we are using this non-chemical approach or this non-chemical biological microbial approach for agricultural waste decomposition? Because they have abilities for rapid growth and multiplication abilities or activities, expression of multi-enzyme complexes which jump on these agricultural wastages for their degradation, then ability to colonize diverse environmental niches. They can adapt to different environmental niches. So you can collect the CDMs from every environments, like you can collect them or you can isolate them from cow dung, you can isolate them from decomposing wood, you can isolate from degraded soils, you can isolate from any kind of soils, but population may vary, but you can isolate from different substances. Okay, vermicompost, you can isolate them. So to develop efficient nutrient management practices or packages, this is a very important need of quality organic manure. And as a result, in addition to this, they play also important role for disease protection, because we have also established some cellulose degraders, okay, which have their potential abilities in nutrient management programs. They have also some potential their works for disease suppression. Okay, so next please. So lots of diseases has been under now our control, or we are using these CDMs for controlling some of the T diseases. These are also in our experimentations. So this is another thing, you see this, you know also this, I think these are potent earthworm species. So these are usually available in our northeast India. Okay, Yesenia fetida, we have these strains, Paranex, then Amanthus, then Dravidia. So we are using these microorganisms along with these earthworm species, and we have got that co-inoculation of these earthworm species along with these CDMs. They works more. Okay, so enormous prospects in using CDMs for agro-waste bioconversion, microbial fortification, that means addition of these beneficial microorganisms, these cellulose degraders for soil health, crop productivity, and eco-enterprising. Additional enrichment of the compost with this compost, which is prepared due to the degradation of this agricultural wastages. They can also be enriched with the addition of some important botanicals, then humic acids, amino acids. So that means their quality can also be enhanced with the addition of certain substances. So they can be then again applied it for application in this degradation. Next, please. Yes, vermicomposting can be applied for, and this is one of the student asked one question, that vermicomposting can be applied for CDMs. Yes, CDM and earthworm species can be used. That means they can be co-inoculated together. Okay, they can be inoculated together for compost preparation. So this is one of our own experiments which we have published in 2019, where we have prepared a very low cost organic manure production. Yes, one of the important thing you should keep in mind, or we should keep in mind while doing this compost preparation, is that it should be cost effective. No? So everywhere, if we do cost effective manner, it will be acceptable by the farmers. Okay, so globally it will be accepted. Otherwise, as we know that chemicals are very costly, so we have to do for some other alternative practices like microbial approaches for low cost organic manure production. So we have done peat preparation, we have collected the weeds, we have sopped them, we have dried them, set dried them, and we have done lots of exercises for saving them. Okay, so then we can do their commercialization purposes. Next slide, please. Yes, for sustainability is very much more essential. Then significance of microbial decomposition. Yes, one of the important parameter is that you have to, after preparing this organic manure, no? Organic manure, that should be a quality. What do you mean by quality? There should be some indicators. Yes, so there are some indicators by means of which you can judge the quality of your organic manure production, which you have prepared by decomposing, okay, these agricultural wastages. Visual screening, this is one of the important parameter, that physical observation. So physical observation for temperature, compost should not be very heat, it should not be heat above, means beyond 20 degrees centigrade. Okay, so their color should be between dark black and then brownish. Okay, so then odor, it should not be very offensive odors, no? So other impurities, it should be free from other impurities like any kind of heavy metals, emerging contaminants. So there are lots of laboratories which can test these impurities. Okay, so moisture content, this is one of the important parameter which need to be seen while preparing the compost. So one of the important tests is squeeze ball test. The compost you should hold within your, means hand, within your palm. So that is a squeeze ball test, like a squeeze ball. Then bulk density is one of the important parameter. It need to be carried out, or it need to be carefully screened, because porosity and moisture content of the compost, it should be a very reasonable limit. Otherwise, if the porosity is very low, it will not work in soil, because it will again make the soil compact, na? So that's why porosity and moisture content judgment, or their regularity maintained, is very much more essential. This is important parameter for quality compost. pH, you know, everywhere, every agricultural system, they have a standard pH. Like in tea soil, tea generally grows in acidic pH, no? 4.5 to 5.5. That's why you have to use the compost in that pH only. Then organic matter screening, major and secondary nutrients. Besides, you have to go for testing. That means status recovery. After the preparation of the compost, we have to go for nutrient recovery percentages or compost recovery percentages, because until and unless they are rich in nutrients, we have no use to use them in agriculture. No, there should be there should be enhancement in nutrient status. That's why we are using this compost or this type of quality manures, and they have been highlighted over this chemical applications. Then cation exchange capacity, we have to screen out. Compost stability, it is one of the important parameter. So the compost you prepare, that should be stable. That means they have to be active with beneficial microbial activities, and self-heating is one of the another treatment. So they have to be incubated, okay, and their temperature should not go beyond 20 degrees centigrade. Then it should be tested for different impurities, contaminants, heavy metals, and emerging contaminants, and that should be, and the compost should be free of any kind of contaminants. Next slide, please. Yes, we have learned that compost have lots of advantages, or this agricultural decomposition of these agricultural wastages by using this biological agents, it is very much more beneficial. Why? Because they can supply plant nutrients and increase nutrient uptake. Naturally, they are rich in diverse nutrients like different kinds of macronutrients, vitamins, then enzymes and antibiotics. Improve soil physical properties like structure, texture, water holding capacity, etc. I am not explaining it by our own. We have done the experiments. Okay, I will show you some results. So after the application of this compost, they can increase the rootlet size and longevity. Then they can also make the plants more resistant against different kinds of phytopathogens. They can improve the water relations in soil and make the plants more drought resistant. Then enhance different kinds of plant growth promoting activities, as a result of which the plant can show their vigorous, their growth and development is very good against control. Then enhance phenolic activity, salt and heavy metal tolerance. Then again, the carbon dioxide which is released due to this composting, they can also be treated as a important carbon fertilizers. Okay, so they can be trapped. Next, please. So there are lots of advantages. These are also in addition to these advantages, these agricultural waste decomposition products, that means bioconversion of agricultural wastages and then preparation of this compost, they can be a useful component of INM. We are regularly using it in INM technologies, means integrated nutrient management approach, where we have to do maximum exercises for non-chemical approach of what agriculture management. Okay, so it functions in long duration, then production cost is much less than the chemical fertilizers, and these application of this quality manure which is coming from this bioconversion means experiments or bioconversion procedures, they are environmentally safe, eco-friendly, and boast protection against diverse soil disease, and then it can also act as a biocontrol agents. Okay, so they can be also applied it in our experiments. We are using them as an important component of IDM, integrated disease management. Next slide, please, ma'am. So this is one of our important publications in 2019 from ICR. So where here we have established that with the application of this compost, we are getting more amount of nutrients, more amount of enzymatic activities, and compost recovery percentage. Next, please. Against control. So this is the first one, analysis for microbial populations. You will be surprising to know that lots of microbial populations like cellulose degrading microbes, phosphate solubilizing microbes, nitrogen fixing microbes, total bacterial fungal populations, they are getting enhanced due to the application of this co-integration of these biological agents like earthworm species and our efficient means cellulose degrading microorganisms. Okay, so another result indicates that yield and yield contributes. We have done this experiment in groundnut applications. We in our experiment we even we are able to reduce the load of chemicals up to 50%, and 50% reduction of the chemicals is not a joke. It's a very good approach. Okay, so likewise we are also, next slide please, likewise we are applying in different agricultural systems. People are doing experiments with these cellulose degrading microbes. Next slide please. Yes, what experiment, post experiment observation. So it yields maximum compost, nutrient content, beneficial microbial parameters. Next please. So integrated disease management, also it is very much more successful. I have mentioned you that these CDMs, they can also be means act as potent degrading microbes in INM, as well as they can also have their most important potential in disease management in tea, particularly in tea, as we have worked out. Next please. So they are also very much more important for integrated means disease management. So now concept arises for integrated crop management. You have learned about integrated nutrient management, you have learned about integrated disease management. So this is your integrated crop management, where we have to manage the whole environment, okay, in a sustainable manner. You have to reduce the load of chemicals, you have to minimum means you have to say means mindset, or you have your mindset should be sensed to minimize the load of artificial inputs like chemical fertilizers, pesticides, and fossil fuels, and go for some alternative strategies like this sustainability approaches, eco-friendly approaches, use of these different components of this ICM like energy consumption, then set tree management, also another important thing in this type of ecosystems. Then with this compile effort, we can grow a ICM, means integrated crop management practices. Next please. So as you have already visualized that our main aim of this presentation is basically is also one of the important aim, although we are using, now we are also coming short of our time also. So although we are using different CDMs or different means along with its art forms for agricultural waste decomposition for means production of the quality materials like quality organic manure, which can be applied for mulching or which can be applied for organic nutrient practices. No? Our basic theme is also for sustainability production or sustainability crop yield. So for this, we have to cut short the load of chemicals. Escaping the chemical trap is one of the important strategies, as it is a global strategy. Okay, so increasing cost because chemicals are very costly. Unpredicted climate, they are causing unpredicted abrupt climatic changes. Labor shortage are also there. Pesticide tolerance, they are developing pesticide tolerance. So PPC means plant protection code. So this is applicable in case of tea ecosystem because there are some limits of applied chemicals. So we can go beyond those chemical limits. No? So lots of there are different means adverse effects of application of chemicals. Therefore, our aim is to go for some sustainability approaches, non-conventional approach for sustainability development. Next, please. So this is one of our field applications of microbial enriched mulching materials prepared through these CDM activities. So you see in A there are some CDM strains which are ready to jump in the mass production cultures. Okay, so B, they are growing well. These are bacterial mats. Okay, so they have been applied in the C and D. So these are some of the rice straw materials which are getting converted to the bioconversion experiments, and these bioconversion experiments, they have been applied in different systems. Okay, in the E picture you can see these are some of the pruning litters. So that means these are some of the materials which are observed in the ecosystem, and this can be converted. Okay, this can be converted to quality compost with the application of the CDM. We have applied this in the tea soil. Okay, and they have also been applied in the, you see in the photograph, Z and H. So they have been applied as a mulching material in Z, and in H you can see vigorous of the plants in contrast to some of the control experiments where after application of these materials as organic matter in tea ecosystem, so they have a great potentiality to increase the nutrient content of soil. They have a very important potentiality in compost recovery, and overall they can achieve a sustainability approach. With this, next slide please, ma'am. With this, I conclude my today's presentation. If you have any doubts, you can also mail me. So my mail is not visible in the screen because it is in the lower side. So Pranab Bhatta, PRANAB @gmail.com. You can also ask from Madam because she is holding this entire presentations. So you can also contact in the next for if you have any doubts also. So thanks, Madam, for giving opportunity to give a lecture such a with these brilliant students. So thank you very much.
Thank you, sir. So students, we don't have a next session. So if you want to ask any query or questions regarding your projects, the
Yes.
floor is open.
Yes.
Anything, any question on microbiology? Somebody has raised the hand. Chakriti
Yes.
Bhattacharya.
Yes.
Yes, please go ahead.
Yes, please. Yes, thank you very much, Moiri Pegu.
No questions?
Students, if you have any queries, please. And
Hello, sir. Hello, sir.
yes,
This is
yes,
this is Dr. Swati from Srishti, sir. I
yes.
just wanted to ask one question, sir.
Okay.
Sir, you told that metagenomics is also working for this understanding the soil diversity.
Yes,
Generally, we heard that metagenomics is used in the area where extremophiles are there, where
yes.
we can't where we can't like we won't able to culture the bacteria. There we are using the metagenomics approach. So how in this particular field we are using metagenomics, sir?
Actually, metagenomics, we can use this technique of means biological, modern biological tools and techniques for isolation for or extraction of the direct DNA extraction of means these microbial genomes. No? So we
Yes,
can
sir.
trace their phylogeny. So if we know what type of microbes are there in our environment, we can easily trace them, and we can easily work with these microbes not only in the decomposition activities but also in other beneficial activities. So that's why this field is now emerging in a field. But in case of farmers related or oriented means system, we have also advantages in culture dependent approach. That's why my today's presentation is basically concentrated on isolation and then collection and then direct field application, actually, because direct field application is basically beneficial for farmers and our friendly means planters and whatever they are engaged in our agricultural production system. So that's why.
Okay. Okay, sir. So we can use metagenomics in
Yes.
normal environment also. It's not needed
Yes.
that we have to use in extremophiles or extreme
No.
conditions
No.
where
No.
we
No.
can't able
No.
to culture the bacteria.
Yes.
So, but in metagenomics, we are going to get n number of bacteria. Like diversity is very vast.
Yes. Yes.
So in that we can aram se use it.
Yes. 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 We can trace their means diversity and we can use them properly.
Okay, sir.
Yes.
Okay, sir. Thank you so much, sir.
Thank you very much.
Student, any other questions you want to ask? You can go ahead and ask.
Somebody is asking chat box that how can we lower the pH of compost?
How can we?
How can we lower? How can
Yes, so pH for pH management, we have to do some soil amendments. If we go for some not means non-chemical approach of means pH management, we have to go for some soil amendment means structures like I have mentioned some like lime. Na? So lime we can use for means lowering the pH or this type of activities. So in addition to this chemical approach of this pH management or whatever, we can go for some non-chemical approach for sustainability development of pH management. So there is no problem. Okay, and you have to actually for I have already shared a screen on these indicators of compost management or indicators of good quality compost, no? Where I have mentioned that visual observation is very much more important because you have to see the odor, you have to see the color, and you have to taste the pH. And for pH management, you can see, you can use the soil amendments properly. There is no problem.
Okay, thank you, sir.
Okay.
Thank
any other question?
So there are 50.
Sir,
Yes, yes.
one more question. Any tips for lowering EC? Like in a FCO regulation, the EC should not be more than 4 DS per meter, right?
Okay.
Decimals per meter.
Electrical conductivity.
So, yeah,
Elect
electrical conductivity.
yes,
So,
yes,
how
yes.
can we lower that in a compost, specific compost, like you say, biodynamic compost or vermicompost, if it is more than 4 decimals per meter?
So you
So, how
see,
can we lower that?
you see a quality compost, it have different parameters, no? So different indicators. So you have to go for their visual observations, you have to go their electric conductance, and you have to go there lots more things, no? So you have to actually, this actually depends on your composting activities. You can't give any additional chemicals for means measuring this or for controlling this EC, no? So you have to go, or you have to standardize your actually bioconversion experimental this composting means yeah, period, no? So incubation period. So it have to be standardized actually. So if you standardize, if you can standardize, normally composting takes place after 45 days of your composting. Okay, so for some agricultural use purposes. Otherwise, your pot experimentation you can also see from 30 days. Okay, so from 45 days up to 90 days up to 150 days. So it will automatically come to your actually necessary if you use proper CDM strains, and if you done these CDM strains in a proper way in your lab testing, then there will be, I think, no problem. Obviously, we have not got yet any kind of means any problems related to ECs. So normally this issue get resolved if you do standardization in your procedures, and if you keep the composting procedure in a very means this period of incubation, it depends actually. So there is, I think there is no problem. We have faced it last 4, 5, 6 years. Okay, so there is no issues, I think. Thank you very much.
Okay, thank you, sir. Sir, you mentioned about pH, there is liming, but my
I have
pH in a, yeah.
okay, okay.
So my pH is in a basic. So liming is
What?
for acid, I guess. Yeah.
Oh, no, no,
You,
no, no. Actually,
yeah.
I am providing some soil amendments. So not actually, I am giving that pH can be adjusted by using some this type of soil amendments. Okay, so we can adjust the pH. There is no problem actually, because we are doing the bioconversion experiments. Okay, so there is no problem. Okay.
So no more questions, students.
Thank you very much, Saif Afridi Hussein.
Yeah. So,
There are
sir,
some-
I'll share your email ID with students.
Okay, okay.
And
No problem.
yes, sir. So,
Okay.
sir, thank you so much for your detailed presentation, and
Okay.
it was really informative.
Okay.
And I hope students will get some insights from your
Okay.
presentation for their projects.
Okay, ma'am. Ma'am, may I request one thing that since in the presentations, we have also